Hey, good morning, everyone. So I want to start off today with the bonus question from last time, which was why do we have so many copies of MHC proteins and why are they so polymorphic in the population? There's obviously an evolutionary advantage for that. It helps with our survival. And if you think about it, there's two different reasons. One is each individual has multiple MHC genes and they're codominantly expressed. And so that increases that one individual's chance of being able to present a peptide from a pathogen, having the right anchor sequence, so that you, for anything that comes in, it's very likely that some of the T cells will be able to recognize that pathogen peptide on that MHC. And so you really have 6 chances for MHC1 and at least 6 chances for MHC2. One little secret is that you have 6 different MHC genes, but there's actually, for MHC2, there's an alpha and a beta. And the alphas can kind of pick and choose which beta they want to pair with. So sometimes you can get mixing and matching. But you have at least 6 different chances for MHC2. So that really enables any given individual to be able to make a response to infection. Now, the second question is, why is it so polymorphic in the population? Right? Why do we have so many different versions in the population? Well, that means that if you have a population of people and let's say you don't make a response, then that person may die, but most of the people will have a good chance of that. So you have lots of different copies or lots of different versions of those copies so that at least some people will be able to make a response. And so that protects the population. Right now we have something like 19,000 different MHC1 alleles and 7,000 MHC2 alleles that are known in the human population, which is quite a lot. Now there are other answers for this that you could justify, saying polygyny increases the likelihood of T cells being selected for, so it's a more efficient process, but in reality, it's mostly just to protect us. Okay, so last time we went over the T cell receptor structure and a somatic recombination to get a T cell receptor. Now we're going to tie that into T cell development because it's very similar to B cell development where T cells rearrange the T cell receptor, then they test. Then they rearrange it again, and then they test. And so, but there's an additional step for T cells. So let's all sort of go back to where we started, right? T cell progenitors come from the bone marrow. And so they make a decision in the bone marrow to become a T cell. They then enter the bloodstream and go to the thymus where they complete their maturation. Once the T cells undergo all of these processes, they then exit the thymus into circulation. Okay, so it might not surprise you. We've talked about how do T cells get into lymph nodes, and it's via these high endothelial venules, these specialized post-capillary structures. So they're on the venous side, which is low pressure, that have lots of invaginations and express the right ligands for T cells to stick there and get into the thymus. So the thymus, just like a lymph node, has high endothelial venules. Once the T cell progenitors go into the thymus, they undergo somatic recombination to express this semi-random T cell receptor. So this fits with the clonal selection theory that each T cell has a unique T cell receptor on its surface so that if you are infected, some of those T cells can recognize whatever is infecting you and make a response. Now for B cells go from the immature to mature phase when they undergo negative selection, but T cells have to also be able to recognize MHC, and so they have to undergo positive selection that yes, they can see MHC, and then negative selection that they don't recognize self-peptides on that MHC. And so it's a more inefficient process than B cell development. But these selection processes are what we've talked about so far as central tolerance. This is deletion of self-reactive cells. Now, when we're during the lecture, I'll actually point out where We sort of skip this part for some cells. Once they undergo this, they then exit thymus via the high endothelial venules. They go right into the circulatory system, where they start this migration pattern between the lymph and the blood. So they spend most of their time hunting around lymph nodes for antigen, but they're essentially circulating T cells. So let's talk about the T cell progenitors. At this pre-T sort of version of a T cell has to make a decision, or a common lymphoid progenitor has to make a decision, am I going to become a T cell or a B cell? And it turns out that the default pathway is to become a B cell. So there is a protein that's expressed in the thymic stroma that's called Notch, or Notch ligand, which signals to notch on the T cells. And if the developing lymphoid progenitor gets a signal from this Notch protein, that tells it to become a T cell, exit the bone marrow, and go to the thymus. If it doesn't get it, it becomes a B cell. So it stays in the bone marrow, undergoes somatic recombination and negative selection to become a B cell. The T cells, on the other hand, that get this Notch signal, exit the bone marrow, they go into the blood, and eventually enter the thymus through the high endothelial venules. This is where they complete their maturation cycle to become a mature T cell. So what happens if you don't have a thymus? Well, it's actually a fairly common primary immunodeficiency called DiGeorge syndrome. And this is patients who have a defect in tissue differentiation during fetal development. So these are sort of global transcription factors. And it doesn't just affect the thymus, it can affect the most common thing is that they need a heart valve repair, but it can affect their heart, their thyroid, it can affect facial development. So DiGeorge syndrome patients typically have a cleft lip. And so that needs to be surgically repaired, but they'll also lack a thymus. And so what happens is you'll get normal B cell development because B cells don't need a thymus for development, but they don't have any mature T cells. And we call this a T negative SCID, or severe combined immunodeficiency. And what happens with these patients is typically you can cure them by, first of all, repairing the other development defects by surgery. But if you implant a thymus into these patients, then their T cells then have a place to mature, and you restore the T cells. And so it's a curable disease. There's a mouse model of this called nude mice, and they have a defect in this global transcription factor FOXN1 that essentially makes them not have hair, and they don't develop a thymus. And there are other defects in these mice similar to a DiGeorge syndrome patient. And again, they have normal B cell populations, but no mature T cells. And so they have this severe combined immunodeficiency. And you can cure it the same way that you cure a DiGeorge syndrome patient. And in fact, nude mice were used to explore how do you cure that. So a question that I have for you is, why is it a severe combined immunodeficiency if they're only lacking T cells? Anybody can chime in and answer.
Is it because the T cells are the controllers of the immune system and we have our CD4 T cells?
Right, so the idea is that for a fully functional B cell response, you require those checks and balances of having a CD4 T cell response that provides that full signal saying, yes, there's something here, let's make a full response. And so if you don't have T cells, you don't have T cell help for B cells, and they can make some antibody responses, but they're by and large defective, right? Because the T cells are directing what kind of isotope or isotype of the antibody is made. They're inducing hypermutation and affinity maturation, so you get a better antibody. But if you don't have them, then none of that happens. Okay, so how do we prove this? Well, there's two versions of SCID mice, right? You have RAG2 deficient mice. And remember, RAG2's job is to cut the DNA during somatic recombination. And so if you don't have RAG2, you don't have T cells or B cells. On the other hand, you have nude mice that don't have a thymus, so they don't have T cells, but they have normal B cells. And so if you, just get my marker out here. So if you take bone marrow from a nude mouse and put it into a RAG2 deficient mouse, then you get repopulation of all the cells. So it means that the bone marrow in a nude mouse is fine, right? There's no defect in the T cell progenitors. So you get normal B cell and T cell development. Now, on the other hand, if you take a thymus from a RAG2 deficient mouse and implant it into a nude mouse, now you will restore T cell development in the nude mouse, which means the only thing that they're missing is that thymic environment. Now, the thymus, or the thymic sac, is not derived from bone marrow, but you do need it for normal maturation of those bone marrow derived T cells. So this kind of experiment put the nail on the coffin, saying, yes, in fact, the thymus is absolutely required for T cell development. So what does a thymus look like? Well, first of all, it's arranged into many lobules or lobes. Lobules are just little lobes that are separated by trabecula. Trabecula are these long sort of endothelial layers that separate the different parts of the thymus from each other. But in reality, it is really separated into the cortex and the medulla. The cortex is sort of, in cartoons, we draw it as the higher region, and the medulla is the lower region. But in reality, it's essentially the medulla is surrounded by the cortex. And you can think of the cortex has one job, and the medullary region has a different job. And the cortex is where somatic recombination occurs and where positive selection, meaning that developing T cell can in fact recognize MHC. In the medullary region, that's where negative selection occurs, where your T cells are being selected against anything that recognizes self antigens. Now, this process is really inefficient, okay? And these numbers are sort of estimates, but it's difficult to actually get a real count on them. They do pulse chase experiments where they're trying to see how many T cells are developing in the thymus and then how many actually exit the thymus. And so you get about 1 million T cell precursors from the bone marrow entering the thymus each day. Now that's about 1% of the total population in the thymus, okay? So there's mostly pre-T cells in the thymus. That's about 100 million. During that time, these cells will proliferate, okay? So they're undergoing somatic recombination, and just like B cells, if they get a beta chain, they undergo proliferation. But then they have these selection processes. They have positive and negative selection, so only about 2% of those cells actually make it through that process and exit the thymus. So it's a very inefficient process, and this was first noticed by looking at macrophages in the thymus, which were called tingible body macrophages. And this is using the eosin dye, right? So you have hematoxylin and eosin to stain cells. And what they found was that the macrophages in the thymus had lots of fragments of nuclei in their cytoplasm. Okay, all of these dark purple circles, that's actually fragments of other cells' nucleus or nuclei. And so they called them tingible body macrophages. Why those occur is that those macrophages in the thymus are engulfing all of the cells that are dying. And it's so rapid that the macrophage doesn't have time to completely digest everything before it's taking up another cell. And so it's a very inefficient process. Why is it? Well, okay, so if we just take what limits the cells that can come out of it? Well, the first is you got to rearrange the T cell receptor in frame. That's a fairly inefficient process. We saw that for B cells. The second thing is it has to pass this positive selection, and then it has to pass negative selection. So it can fail at any of those three stages. And so the 98% of cells that are undergoing apoptosis because they failed one of those three things are phagocytosed by macrophages. And that's why we get these tingible bodies. They're just incompletely digested nuclei because they're constantly eating up these dead cells.
Dr. Blackman,
Yes.
I'm kind of confused of how there was the same 10 to the 6 precursors T cells going into the thymus and 10 to the 6 mature T cells exiting the thymus being inefficient.
Well, because if you think about it, these 10 to the 6 cells are undergoing proliferation, and we'll talk about that. It's essentially the double negative 3 stage. So they're proliferating. And then they rearrange the alpha chain, and then you have to select against that population. So there's a step where they come in, and they're rearranging the TCR beta, and then they proliferate, then they rearrange the alpha, and then they have to undergo these other processes. So even though you have 10 to the 6 precursors coming in, after this proliferation, it's 50 times higher. So that's where the inefficiency comes in.
Okay.
So as I alluded to, there are different stages of T cell development. In order to talk about them, we need to talk about flow cytometry. Flow cytometry is, as the name would suggest, just measuring cells. Cytometry is cell measurement in a flow-based system. And so what you do is you take antibodies to the different T cell markers. So you can have antibodies to CD4, which we've talked about, or you can have antibodies to CD8. Okay, and so those are the two main markers that we're going to use. In the thymus, you can have cells that have neither of those. You can have cells that have both, or you can have cells that have one of them. And so what you do is you use these antibodies that are labeled with a fluorescent dye, in this case, fluorescein isothiocyanate or Texas red. Those are just two common dyes that are attached to these antibodies. And you pass them through a laser in a flow-based system. They're flowing through the laser. And as the laser hits those dyes, you're capturing a signal for that one cell. Is it positive for the green signal, or FITC? And is it positive for the red signal, which is Texas red? And so you do that for several thousands of cells, and you can come up with different populations. In the thymus, everything starts off here in the double negative stage. This is where you're essentially doing somatic recombination. They then transition to the double positive stage, where this is where you would undergo positive selection, and then ultimately they'll go from there to either the CD8 single positives or the CD4 single positives. And that's where they undergo negative selection. Now, if you notice, that's what a thymus looks like. In a lymph node, you don't see those cells. You see single positive CD4 cells or CD4 or CD8 positive single positive cells. And if you're looking at lymphocytes, what would these cells be? The double negative for CD4 and CD8. Any ideas? They're lymphocytes, but they're not CD4 positive and not CD8 positive.
Naive.
They're not T cells.
B cells?
They're B cells, okay? So you have two T cell populations and one B cell population. The ones in the thymus are not B cells. Those are T cell progenitors. Now what this looks like in reality is a little different. I mean, it does look like you have these different populations. Here we have very few single positive cells. Most of the cells in the thymus are double positive, okay? And that's because this is where you have the most stringent criteria. They have to have a TCR on their surface, and they have to undergo positive selection. And then you do have populations of single positive cells, okay, that are mature T cells that are going to then exit the thymus and go start looking for antigen. When you look in a lymph node, you really don't see any double positive cells because they don't make it out of the thymus. You have CD4 cells and you have CD8 cells, and then you have other cells, including B cells and then some granulocytes. Okay, so when we're talking about thymocytes, we're going to be talking about double negative, double positive, and then single positive. And we'll divide the single positive into CD4 cells or CD8 cells. So all of the T cell precursors that enter the thymus start off at this double negative stage. Okay, they don't have CD4, they don't have CD8, they don't even have the signaling proteins that are the CD3 proteins, right? And this is the stage that they begin to rearrange their TCR genes. Remember, they rearrange beta, gamma, and delta first, all at the same time. Then this is going to determine, is it going to become a alpha beta T cell or a gamma delta T cell? They're also beginning to express the pre-T alpha protein, and this will come into play a little bit later. So now if it rearranges the TCR gamma and delta, if both of those are successful, it essentially remains a double negative cell, exits the thymus, and they go out into the periphery. Gamma delta T cells do not undergo negative selection, and this has to do with how they recognize antigen. Their gamma delta T cells have much less diversity than an alpha beta cell. They don't have junctional nucleotides that are added, so they're basically coming with a preset receptor for something that you don't have, some structure. And they sit out in epithelial tissues just waiting for something to come in and recognize it. So they're much more like a natural killer cell. Okay, and we don't go a lot into their biology in this course because it's sort of, I want you to understand basics of immunology before we start looking at the oddballs of immunology. Okay, so that's what happened if you get gamma and delta rearranging. But what if you get beta rearranged productively? Now remember, gamma and delta, you have about 11% chance of being productive. It's a 33% chance for gamma and 33% chance for delta. For beta, you have about a 33% chance. And so you actually get more TCR beta rearrangement productively than you do gamma delta. And so these cells will express the beta chain, but they'll express it with a surrogate. In this case, it's the pre-T alpha. This is just a structural protein. It doesn't actually encode an antigen receptor. And this is what tells the cell, okay, yes, you should keep going on to rearrange the alpha chain. And so you can see it's sort of like the same thing that we went with B cells. You rearrange the heavy chain for B cells, and you express the V pre B and lambda 5, the surrogate light chain, and then you test. Did I get something on the surface? Did I get a protein on the surface, which means that it was a productive rearrangement? If it's yes, it keeps going. If it's no, it's not entirely clear if you get another chance for T cells. T cells are a little more selective. So now these cells that get a positive signal will rearrange the TCR alpha gene. And this is where they then will continue their maturation and they become double positive T cells. So they'll express both CD4 and CD8. And when they do this, now they undergo this process of positive selection. And we'll talk about that a little bit more because these are actually 3 different steps. There's positive selection, there's lineage commitment, which one are you going to become, a CD4 or a CD8 T cell? And then there's negative selection. That's all happening, this transition between the double positives to become the single positives. But once you have these single positive cells, they undergo negative selection and making sure that they don't recognize any self-antigens. And if they finally pass that, then they can exit the thymus into the blood and start looking for antigen throughout the lymph nodes. So if we break down the steps here, I think T cells are a little easier to remember in terms of which things are happening, because we don't call them pro and pre-B cells, we call them double negative 1, double negative 2, double negative 3, and then double negative 4, and then double positive. Okay, so they don't express CD4 and CD8 until they get to this stage. And then they have to choose which kind of cell, and they become either CD4 cells or CD8 T cells. But essentially, you can think of these as the same steps are occurring. There's some slight differences in timing, but for this course, we're going to sort of homogenize it so that it's easier to see that they're the same thing. Okay, so somatic recombination is happening at the double negative 1 and double negative 2, the same as it's happening in the early pro-B and the late pro-B. And then there's a test, right? To become a pre-B cell, you have to have the heavy chain expressed with the surrogate light chain. For the T cell receptor, you get a TCR beta expressed with the pre-T alpha. And that's how you know that there's a signal. If it passes that, then it undergoes somatic recombination again. For B cells, this would be at the small pre-B stage. For the T cells, it's at the double negative 4 stage. And that's where for B cells, you're rearranging the light chain. For T cells, you're rearranging the alpha chain. And then again, the next step is to test. For B cells, that's at the immature B cell stage. And for T cells, it's at the double positive stage. And so that for T cells, it's actually a signal, do I get to keep going? And then to become a mature cell, for B cells, they have to undergo negative selection, where T cells have to undergo positive selection and negative selection, as well as this choice of, do I become a CD4 cell or a CD8 T cell? Okay, but they're actually very similar to each other. There's a little bit of a difference here, though, in that you can get different versions of T cells. For B cells, they're, for the most part, in an adult mammal, they're fairly homogeneous in their steps to development. But you can see for T cells, there's a lot more forks in the road. And the first fork is that as at this double negative 2 stage, right? So during the double negative 1 and double negative 2, you're rearranging beta, gamma, and delta. If you get gamma and delta, those cells will become a gamma delta T cell and exit the thymus. Okay, so that's the first fork in the road. If they get a beta signal, then they'll continue down this pathway towards the double negative 3 stage. And then just like a pre-B cell, right, the large pre-B, it means it's blasting or it's undergoing division. That's what cells do to divide. They increase their cytoplasm so that each cell can have enough. Double negative 3 cells also undergo this proliferative stage, okay? And so there are essentially the same things happening for the T cells as there are for the B cells. You're getting half of the receptor expressed with a surrogate chain or with a chaperone protein that allows that to be at the cell surface. And then it has to undergo rearrangement again. So for T cells, it's TCR alpha. And then there's another step of, okay, do you have something on the surface? Can you recognize something? Now for B cells, we said it was this sort of tonic signal or a static signal where it's cross-linking the B cell receptor, right? At the large pre-B stage. In an immature B cell, we're not entirely sure what the ligand is or if there even is a ligand. Most people think it's sort of just you're expressing the receptor and you're getting enough of a signal that B cell knows to go on to the next stage. For T cells though, they have to be able to recognize self-MHC. Okay, so they're going to have a TCR alpha and a TCR beta on their surface. They're double positive now because they're expressing CD4 and CD8. And so they then undergo this negative selection phase where any self-reactive cells get deleted. This is true for B cells as well as T cells. Now for B cells, remember when they become mature cells, they'll start also expressing the IgD isotype of that same B cell receptor, and that's RNA splicing. So a mature B cell will have IgM B cell receptor and IgD B cell receptor on the surface. It's the same variable rearrangement, right? So they recognize the same thing. It's just a way for mature B cells to have more on their surface. T cells don't do that. They don't just stick with the T cell receptor. They don't have an isotype, so there's no way for them to do another type of T cell receptor on their surface. Now there's this other step here. B cells will just exit and all the B cells are the same. But T cells also have this fork in the road called lineage commitment, where they have to decide, if I recognize MHC2, I should become a CD4 cell. If I recognize MHC1, I should become a CD8 T cell. And we'll come back to that in just a second. But if we look at this picture, this first break in the road is TCR, beta, gamma, and delta, right? And they're all rearranging at the same time. So what if you got all three of them rearranged? What would happen? Well, it turns out the gamma delta signal is dominant here. And that's partly because it's a lower frequency. So you start off with all three of those genes being rearranged at the double -1 and 2 stage. If you get a TCR gamma delta on the cell surface, this is a very strong signal, and that stops TCR alpha beta rearrangement and forces that cell to become a TCR gamma delta T cell. If you get a TCR beta expressed on the surface with the weak or with the pre-T alpha, that generates a fairly weak signal, and that says, okay, we're going to stop gamma delta rearrangement and then go and rearrange the TCR alpha. So it's really, if you get a gamma delta TCR, that's what happens. It doesn't matter if the beta is going to happen, because you can imagine this is a strong signal. Adding an additional signal to it still makes you go this way, okay? But if you just have a weak signal, then you can go this way. Now at the double -3 point for the alpha beta T cell pathway, they're going to downregulate the RAG proteins, and that stops recombination. And you're testing for the receptor, but then if they see that, then they will undergo proliferation and keep going. So the strong signals from the TCR gamma delta, though, prevent re-expression of the RAG proteins. Okay, so RAG1 and 2 that are central in somatic recombination, that stops them and doesn't allow them to be re-expressed later on. So gamma delta T cells are done, so they exit. But if you get this weaker signal from the TCR beta and pre-T alpha, then those cells will turn back on the RAG proteins to rearrange the TCR alpha, and then hopefully get a productive rearrangement there and go on to become alpha beta T cells. Okay, so let's go through this in the different stages of T cell development, because it looks very similar to B cell development, with the exception of there's some differences in the rearrangement of these genes. And we're going to focus mainly on TCR alpha and beta, just because those are the classical T cells and the ones that we're going to talk mostly about in this course. Okay, but remember, gamma and delta are both rearranging first. So the initial stage at the double -1 is to start the D to J rearrangement for the beta chain. And you get to the double -2 if that's rearranged, and you start the V to D to J. Okay, so the first step is to take these two together. Once those are rearranged, in this case it went to the third one here, then you're going to select a V gene and join it to there. And that's at the double -3 stage, where you have the TCR beta is rearranged, and it's expressed on the surface with the pre-T alpha. And this is your test. Did you get something on the cell surface? If you do, then those cells will proliferate. So double negative 3 cells are large, just like the large pre-B that are proliferating. And how do we know this pre-T alpha is important? Well, the experiment is to take a RAG2 deficient mouse. So that mouse does not have any T cells or B cells. And you put into it bone marrow from a mouse that is knocked out for the pre-T alpha, but expresses a transgene that has a fully formed or fully rearranged TCR alpha, okay, which should be able to pair with any beta that is expressed. And so the bone marrow cells will give rise to T cell progenitors. They go to the thymus. And the question is, do you need the pre-T alpha or is it just a placeholder, right? Could any alpha chain work in that situation? And the answer is no. You get no T cells developing or very few T cells developing, which tells you that replacing the pre-T alpha with any TCR alpha is not okay, okay? And the reason I think most people think is that if you express a fully functional TCR alpha, you're delivering this strong signal. They're actually going down the the gamma delta pathway. You're not getting alpha beta T cells. Okay, so you need this pre-T alpha. It's not just a placeholder. So how does this signal? Well, it's still debated, okay? We don't always know how these things are working. We just sort of know what the proteins are that are important. And so you have the pre-TCR alpha, or pre-T alpha, binds TCR beta that's being expressed, and it stabilizes on the surface. The cell is also expressing CD3 proteins that you need to get a signal from that T cell receptor. And if it gets that signal, it stops everything from rearranging, saying, okay, we got something on the surface, stop trying to do anything else, this is the winner. The second thing that happens is those cells will proliferate. And just like a B cell, it's essentially saying the T cell is saying, I got something on the surface, I'm going to proliferate and maximize my chances of getting a TCR alpha that works with this TCR beta, okay? So there you have several chances at a good TCR alpha. So that's the whole point of the proliferation. If you rearrange the alpha chain and then express both a fully rearranged alpha and beta on the cell surface, that's really the double negative 4 stage, where they then will again test for this, and if they pass the test, they can go on to the double positive stage. Okay, so the double negative 4 stage, right, is where you're really starting the V to alpha, the V alpha to J alpha rearrangement. Okay, you've got a fully formed beta chain, but now you're going to rearrange the alpha chain, and here we're going to jump between these two. Now remember what that does to the delta loci. It's going to cut it out, right, because it's in the middle of the TCR alpha. And so this is another way that you ensure that cell has to choose between alpha beta versus gamma delta. Now, if we break down the double positive, there's actually 2 different stages here. There's an early double positive stage and a late double positive stage. But the early double positive stage is where you're getting surface expression of fully recombined TCR alpha and beta on the cell surface, okay? So that's the first time that you're going to also see CD4 and CD8. At the late double positive stage, this is where we're actually going to test for recognition of self-MHC. And this is typically just called positive selection, meaning, yes, I'm able to recognize my own MHC. If I do that, then I can go on to the next step. So there's a ligand for the T cell receptor. So when you look at when we come back to the organization of the thymus, all of these double negative and double positive stages are happening in the cortex. So the double negative cells, or the T cell precursors,
enter the thymus through the high endothelial venules. And these are typically very near the cortical medullary junction. And that's your double negative 1 stage. That's where they start TCR beta rearrangement. They then keep going in the cortex. They're rearranging the, keep rearranging the TCR beta, the V to D to J. And then they're looking for surface expression of the TCR beta and pre-T alpha. And that's the double negative 3 stage. At this point, it's now going to start migrating back towards the medullary region, okay? And so here you're having somatic recombination. You're doing somatic recombination again, and here's your test, okay? Now, if it passes that test, it's going to rearrange the alpha chain and become a double positive cell. So the early double positive is going to be alpha beta positive. And then it's undergoing positive selection. And this is going to occur on cells called cortical thymic epithelial cells. And this is these blue cells here in the thymus. This is the first bottleneck in T cell development. Many cells will develop a T cell receptor that just can't recognize self-MHC. So lots of these cells will die. And that's why you see those tingible body macrophages. If they are able to recognize self-MHC, they head towards the medullary region. And this is where they're going to encounter medullary thymic epithelial cells. Now you can see on this, they're actually interacting with MTECs, or medullary thymic epithelial cells, and dendritic cells. And this was only discovered very recently, within the past couple of years, but both cells can mediate negative selection. And we'll talk about it in the experiments that show that. Essentially what's happening is that these cells are interacting with both, but for the purposes of this class, I want you to remember that medullary thymic epithelial cells are the ones that are mediating negative selection. In reality, both can do it, but it gets a little complicated to explain how both are doing it, and then you remove one and the experiments still work. But in any case, these are the steps, and they're tied to the thymic or the thymocyte migration in the thymus. You're going to the cortex first. That's where you do all the rearrangement. And then you go to the medullary region, which is where you do negative selection. If they pass that, they again exit via the high endothelial venules. Okay, so it's the same exit strategy as the entry strategy. Okay, so what happens, very early experiments in the late 60s, early 70s showed that if you irradiate a mouse, that the irradiated thymus looks different. And essentially what they saw was that the medullary cells would disappear, okay? And for this class, we're going to consider the medullary epithelial cells as sort of a hybrid between the dendritic cells and the MTECs. We're just going to lump them together. And so if you irradiate a mouse, it's basically just left with cortex and no medullary region. And so people use this to try and figure out which cells do what, okay? So irradiation destroys the medullary regions, but not the cortical regions. So the cortical thymic epithelial cells are radiation resistant. The medullary thymic epithelial cells are radiation sensitive. And that's mostly due to these dendritic cells that are there, okay, which are bone marrow derived. In all honesty and disclosure, MTECs are not necessarily bone marrow derived, but I just want you to think of them, the medullary region is radiation sensitive. So if you radiate, you lose medullary thymic epithelial cells. And so you can use this to test which cells mediate positive and negative selection. This is early on in immunology how we figured this out. And so the first thing is the setup for both experiments is exactly the same. It's only what the readout is, okay? And so here's how it works. You take a mouse with the MHC-A haplotype and you irradiate it. When you do that, you're wiping out the medullary region, but the cortical region is staying intact. You then put bone marrow into that mouse that has MHC that are it has both MHC-A and MHC-B, right? Because you have two copies of your MHC, LK and D, in a mouse. And then the difference between the two experiments is the test. All right, in this case, you're infecting the mouse with a virus. And what you're asking is, do I get virus-specific cells that can recognize, or do I get T cells that can recognize virus in both A and B? Okay, so the answer is, they don't kill MHC-A cells without virus. They don't kill MHC-B cells. So you're not getting cells that can recognize MHC-B. They're only killing MHC-A cells. Okay, so what cells have we retained? Well, if we've irradiated this, we've wiped out the medullary region. And so when we put in MHC-A by B bone marrow, the medullary region is MHC-A and B. The cortical region is MHC-A only. Because you don't get any MHC-B cells, those cells have never passed positive selection. So there's no MHC-B on the cortical cells, which are the cells that have to do positive selection. Okay, so the way to think about this is that in this mouse, after irradiation and putting in this bone marrow, you only get MHC-A restricted T cells. You don't get any MHC-B restricted T cells. So the CTECs are the ones that are MHC-A only, and they're radio-resistant. You didn't wipe them out. So the bone marrow is replacing the radio-sensitive population, but they have B. If they were the ones that mediated positive selection, you would have MHC-B restricted cells, but you don't have that. So therefore, positive selection has to occur on the cortical thymic epithelial cells. Those don't have MHC-B. They're the ones that stayed behind after the radiation. Questions on that? I can tell you on an exam, we're going to ask you to describe one of these two experiments. So please, when you're studying for this, draw it out and make sure that you can explain it, being able to explain why are there no MHC-B reactive cells there?
Sorry, Dr. Blattman. So
yes.
They injected the mice with bone marrow, which then replaced the MTC cells, but the MHC-B cells still didn't develop even when they were replaced?
Correct.
And that's because they had radiated the medullary region, so it wasn't able to develop in that area.
They never make it there. MHC-B reactive cells never get out of the cortical region because they have to pass positive selection to go from the cortex to the medullary region. And so
if the cortex is still intact and they can go through positive selection.
But MHC-B reactive cells don't go through positive selection because there's no MHC-B in the cortex.
Oh, that's right. Okay.
Right? So this is the trick. You're asking to go from the cortex to the medullary region, you have to pass positive selection. For an MHC-B reactive cell to do that, you'd have to have MHC-B in the cortex. Well, you don't. So you don't have MHC-B reactive cells passing positive selection. So that's how we knew it had to be the cortical thymic epithelial cells that are important for that. All right, everybody else there? Okay. So what is the point of positive selection? Well, remember how T cells recognize something. They're recognizing this hot dog bun of MHC as well as the peptide in the middle. But if you can't recognize the hot dog bun, there's no point in that T cell going on. Okay, so it's the interaction between the double positive T cells, or T cell progenitors, and the cortical thymic epithelial cells in the cortex of the thymus. And you have to select for cells that can recognize self-MHC. Now, most of the time, those cells can't. It's about 80% of cells can't recognize MHC and they die. Now, it was discovered in 2014 that it doesn't look like T cells have the same pattern of rescue that B cells have. Remember, B cells, if they undergo negative selection or trying to undergo negative selection and they fail, they go back and rearrange the light chain again. They can keep trying to do that. For T cells, it doesn't look like that's allowed. That essentially, we're just going to accept this inefficient process because T cells have to have this more stringent criteria for selection, and they're also far more dangerous. But about 20% of the time, these T cells are recognizing self-MHC. They get that signal for positive selection. They're able to go on. The next thing they have to do is choose, well, am I going to be a CD8 cell or a CD4 cell? And this actually is not as simple as which MHC they initially saw antigen on. It's actually a complicated process because remember, a cell can only test for one thing at a time. And it used to be thought that it was just, well, if it gets positively selected on MHC1, then it's a CD8 cell. If it gets positively selected on MHC2, it's a CD4 cell. But it's not actually that simple because that's two tests at the same time, right? And you can't have both. And so what has been figured out is actually this is an elaborate dance. And it's basically the way the cells orchestrate this so that they're only testing one thing at a time. Okay, so the first step is that your double positive cells, here's our flow cytometry plot of thymic cells, and the double positive cells undergo positive selection. Okay, so that says they recognize some MHC on those cortical thymic epithelial cells. Now the first thing that they do is they downregulate both CD4 and CD8. They become sort of this pseudo double negative population, okay, which is here in orange. But then what they're going to do is continue downregulating CD8, but turn CD4 back on. And so they become pseudo single positive cells. And now they're going to test, do I in fact recognize MHC class 2? If you do recognize class 2, great, keep going, you're a CD4 cell. But if those cells don't recognize class 2, they actually go and start expressing CD8 and stop expressing CD4. So these are single tests and they do it in order. First you test to become a CD4 cell, then you test if you become a CD8 T cell. Okay, so they've committed to one of those two lineages. Okay, now we have single positive cells that have a fully functional TCR alpha and TCR beta on the surface. They've chosen to become a CD4 cell or a CD8 T cell. And now we need to know which cells mediate negative selection. And the setup for the experiment is exactly the same. You take a mouse that has MHC-A, you irradiate it, so you wipe out the medullary region, and you put in bone marrow that is MHC-A by B. So here, the cortex is A, and the medullary region is A plus B. Okay, that's the exact same setup as the test for positive selection. But the difference is the test of that is different. Now you're testing, do I get any MHC-B alloreactive cells? Okay, during positive selection, we know you're only selecting for MHC-A reactive cells, but we know about alloreactivity that some of those, between 1 and 10%, are going to be alloreactive against this foreign MHC. And so the question is, do they reject a skin graft? And the answer is no, they don't. So you've deleted all of the MHC-B alloreactive T cells. So the way to think about this is, okay, we know from the previous experiment, we only get MHC-A restricted T cells developing, right? So you won't get a T cell response against virus with MHC-B. So the question is, do you get any alloreactive cells? And it should be about 10%. So if there are alloreactive cells, this graph should be rejected. So the only cells that we know that have B on them are these MTECs, right? So MHCB is only here. The CTECs only have A. So the cells that are mediating negative selection are the ones that have MHCB on them, and that has to be the MTECs. Questions on that? Right, again, I know these are somewhat complicated experiments. Just remember the setup is always the same. It's just the test that is different. In one case, you're looking for, during that response, did I get any MHCB reactive cells or MHCB plus virus recognizing cells? The answer is no. In this case, you're looking for alloreactivity in a skin graft. Okay, so what's the point of negative selection? Well, the real point of it is to select against T cells that can recognize your own MHC and your own peptides, right? Your cells are always presenting peptides on MHC, and we'll talk about that next week when we go through MHC1 and MHC2 peptide presentation. But the whole point is you want to delete self-reactive cells, because if you don't, you will get severe autoimmunity. And this is between the T cells in the medullary region that are single positive and the dendritic cells and medullary thymic epithelial cells in the medullary regions. Now, it turns out there's actually several outcomes of this. Most of the time, if you have moderate recognition, the cells are like, yep, I'm self-reactive, I'm going to undergo apoptosis. However, there's two other versions of this. One is that if it's a very strong recognition, your immune system actually uses that to generate what are called natural Tregs, or I think the new terminology is thymic Tregs, that basically these are Tregs that are coming out of the thymus with a very strong recognition of self, but in fact their job is to suppress responses. And so they're not going to cause autoimmunity, they're going to suppress anything that comes out and bypasses this negative selection, somehow escapes into the periphery and starts making an autoimmune response. Their job is to suppress it. If there's very weak interactions, that's where we get our sort of conventional T cells, right? They are not self-reactive enough to get them to undergo activation. Excuse me. They're not self-reactive, and so they get to leave the thymus and go out and do their normal circulation. So this is the end of T cell maturation, okay? And you can see that at each of these steps, we're deleting huge populations of cells. So what happens is these cells then upregulate chemokine receptors, in particular, chemokine receptor 7, which allows them to go out to the thymus and start going into lymph nodes. So they'll go into the blood, they'll enter lymph nodes through the high endothelial venules, hunt around for antigen for about 24 hours, and then they'll go upstream to the next lymph node and eventually back into the thoracic duct in the blood. And then spend 95% of their time just hunting around in lymph nodes for their antigen. Now, one of the things that we know, and we've sort of introduced this topic before, is that many of the antigens that they're going to see in the periphery are tissue specific, okay? And this is true for B cells as well as T cells. But remember, there's this partitioning. Naive cells are not just entering your pancreas because they're not just going there willy-nilly. They're actually going from the blood to the lymph nodes back to the blood. And so they never really would see those. But in some cases, you can get damage and release of those antigens. And so you have to have a way of expressing tissue-specific proteins in the medullary thymic epithelial cells so that, pancreas-specific T cells get deleted in the thymus, and they're not bypassing that and get out in the periphery. And so there's this master transcription regulator was identified within the last 10 years called AIR. And it's It was at first identified in patients who have severe autoimmunity. Typically, these patients don't live past a few years old, okay? Because they get progressive autoimmunity to multi-organs. AIR is expressed exclusively in the medullary thymic epithelial cells. And it's basically overrides transcription for a lot of genes, okay? It recruits Pol2, activates it, and then you get transcription of all the genes, okay? So that any gene that could possibly be recognized is being expressed in the medullary thymic epithelial cells, and any T cells specific for those cell proteins gets deleted. In reality, it's not that efficient. You do still get self-reactive cells, so peripheral tolerance is still in place to maintain that your self-reactive responses get shut down. And that includes those natural Tregs that have very high affinity for self-MHC. So when we look at T cell development, this is a review from a figure from a review article we wrote several years ago. We sort of break this down, and I've included a couple other steps here, and just to make sure that you know where they're coming from. The T cell precursors are exiting the high endothelial venules. And then when they enter the thymus, they begin the double negative 1 stage. This is where they're rearranging gamma, beta, and delta. Now for beta and delta, they're doing D to J. For gamma, they're doing V to J. At the double negative 2, they're doing V to D to J. Okay, so double negative 1 and double negative 2 are just somatic recombination. The next step is if you're a gamma delta T cell, you're going to exit. You're done, right? And there's a lower percentage of those, but they get to exit. There's no positive selection and there's no negative selection. Now these cells can become two different versions. They can become T cells or NK T cells. So you have gamma delta T cells. These are very similar to NK T cells that have gamma delta TCR. Functionally, they have slightly different jobs, but they're essentially going out and looking for either disrupted cells or foreign antigens. Now, gamma delta T cells, remember, have very low junctional diversity when they join up the different gene segments, and they're really, that is their selection. There are very few choices, so there's a very low probability of them being self-reactive. They also, their job is to go out and sit in epithelial tissues. They don't move around. So there's very little chance for them to encounter self-antigen because they're not circulating all the time. Okay, but if you get the beta chain rearranged, then you become a double negative 3 cell. You have the pre-T alpha that generates a signal. These cells proliferate, and then they rearrange the alpha chain at the double negative 4 stage, double negative 4 stage. And then if they express the TCR alpha beta, they will then undergo positive selection on the cortical epithelial cells. Now, there's a couple of steps here. In some cases, if they rearrange some of the alpha beta genes, a very narrow set. In fact, in mice, there's only one V alpha that is used by the specialized cells called the NKT cells. It has a very low diversity. Those cells actually escape. And so their job is very similar to the gamma delta T cells in that they're going out and they're looking for aberrant things. And they have very limited recognition. So again, they're not circulating like your conventional T cells. They're essentially going out in tissues and looking for, scanning for aberrant self cells. Okay, so the question is, do NK T cells undergo positive selection? And the answer is no. They skip that. So only your conventional TCR alpha beta cells are going to undergo positive selection. Okay, now they make this lineage commitment here to become a CD4 or CD8 cell. And that's also on these cortical thymic epithelial cells. But as they're sort of percolating towards the medullary region, they have to decide CD4 or CD8. Remember, they're doing that. They downregulate both. Then they put up CD4 and say, do I recognize class 2? Great, if you do, you're a CD4 cell. If you don't, then they turn off CD4 and turn CD8 on, say, do I recognize MHC class 1? And if you do, then great, you go on to the next step. But even after having done that choice, now they do negative selection. So this is on the medullary epithelial cells. And they're testing whether I recognize self-MHC plus self-peptides. And if they recognize it really strongly, you get these natural Tregs. If they don't recognize it really strongly, you get a very weak signal, they become fully mature alpha and beta cells. Okay, so when you're looking at this, where is death occurring? It's occurring here. There's probably some minor death here, but you've already said these cells can mediate, these cells can recognize some MHC to some degree. So it's not really happening at this lineage commitment. They recognize MHC, it's a question is which one. Okay, so there's probably some death, but not an appreciable amount. And then there's massive death here. Okay, this is about 80% of the cells that make it to this point will undergo cell death. And this is somewhere around, it's actually 1% out of 20, so it's about 5% are gonna pass this step. So there's between 95 and 99% of these cells never make it out. And that's why we started to have these estimates of how inefficient this process is. Okay, questions on that? All right. So what do I want you to know? Okay, so first of all, what's the key signal for becoming a T cell versus a B cell, right? Remember there's one, the B cell pathway is the default, but if they get this one key signal, they say, okay, I'm gonna go to the thymus. I think you should know how you can take bone marrow from a nude mouse and put it in a RAG2 knockout mouse to restore the immune system, or you can take a thymus from a RAG deficient mouse and put it into a nude mouse and restore the T cell compartment. Okay, so they can complement each other. You should be able to describe T cell and B cell development and sort of how they match up. Double negative 1 is the early pro-B. Double negative 2 is similar to the late pro-B. Double negative 3 is similar to the large pre. Double negative 4 is similar to the small pre. And then immature B and double positive T cell are sort of at the same stage. Then mature and single positive cells are at the same stage. So a question would be, what is the minimum number of times that a T cell has to undergo somatic recombination? Anybody answer?
Would just be once. Because after the first recombination, then it could, if the gamma delta happens, then it'll go off and it doesn't need to go anymore. But technically, you do still have a T cell there.
Okay, well, even for a gamma delta T cell, it still has at least 3, right? Because it has to do D to J for delta, V to D to J for delta, and gamma. So even though they're all occurring at the same time, it still has to, somatic recombination has to work 3 times in that cell. And it's true for, that's also true for alpha and beta, although it's separated temporally. It's still at least 3 somatic recombination events. But you are correct, it is, I guess they're all lumped together as one, but I'll have to clarify that on the exams. I see that in the chat there's a question from somebody whose mic is not working. Go ahead and type it into the chat and we'll try to come back to that. Okay, so you should know when somatic recombination occurs during each stage of T cell development. And I would say also for B cells. And then being able to tell me what steps of somatic recombination determine whether it's an alpha beta cell or a gamma delta T cell. How do these cells decide to become CD4 or CD8 T cells? And then sort of what positive
I think this
selection
needs
is
labels.
and what is negative selection. Okay, and so during negative selection, what's this master transcriptional regulator that ensures that you have proteins from all of the different organs expressed there?
Dora?
I think somebody's mic is on.
Hi there.
Okay, so the question from the chat is, if we go back to this previous slide, I'm only showing natural Tregs coming from CD4 T cells. And the question is that correct? Well, in biology, there's always exceptions to the rule, but the majority of Tregs that come from the thymus, the natural Tregs or thymic Tregs, are CD4 T cells. There are some Tregs that come out as CD8 single positive natural Tregs, but it's a very low population and it's not really understood how they're working because they don't work the same way as CD4 natural Tregs. And so it's not clear if that's just an artifact or if they have some special role. So the chat question is, what is a Treg? A Treg is a suppressive T cell. Until the sort of late 80s, early 90s, people didn't actually believe in this. Ethan Shivak, basically because they couldn't isolate them and they couldn't prove that they existed. But Ethan Shivak at the National Institutes of Health was sort of the big proponent of this and showed that eventually you could identify these cells, and their job is to go out and if a cell is going to be reactive to self, they're going to shut it down. And we'll talk later about all of the different ways that they do this, but it's a specialized part or a specialized kind of T cell that says, if you're going to be self-reactive, I'm going to shut down that response. If you go back to some of the early lectures, we talked about these when we talked about some of the different syndromes, the APOSED or IPEX syndromes, where if you don't have those, it becomes, you have lots of self-reactivity.
Quick question.
Yes.
So Tregs are developed because they have strong recognition of the MHC and peptide, right?
Correct.
But their job is to get rid of cells that have strong recognition. So do they act against themselves too?
Well, they have strong recognition, but their job is to suppress any cells that are going to react to self. These cells are suppressive, so they're suppressing themselves as well.
Okay.
Or they are ensuring that they do not make inflammatory responses. That's probably a better way of saying it. Okay, so that's sort of what I expect from you on how to prepare from this. Remember, we've got 2 more lectures and then the second exam is coming up, and we, out of compassion, if you want to call it that, we try and make that before the drop dead date of drop class date, and so that you can make a decision at that time. Again, the exams will be the same format of multiple choice, matching, true and false, just so that there's no ambiguous answers. Okay, questions? Oh, and I would say we will certainly ask you about the experiments to show which cells do positive selection and which cells do negative selection. So somebody had a question, sorry.
Oh, sorry. It wasn't really related to much except the DeGeorge syndrome. Does that just happen through genetic mutations? Like if there's someone that has it, will they pass that on? Is it hereditary, I guess is my question.
It absolutely is hereditary. It's defects in transcriptional regulators or transcription factors that are important for many organ developments at the fetal stage or embryonic and fetal stage. They're typically, in mice, it's the FOXN1 transcription factor that sort of regulates our sightedness. And so you often see in people with DiGeorge syndrome is that their stomach is flipped, or they'll have sort of things are reversed inside because the patterning is all off. Now, a lot of DiGeorge syndrome, or most primary immunodeficiencies, result from consanguineous pregnancies, where you're marrying your first cousin or closer. And we'll talk about this later in the semester, but we think that that's kind of very strange for many people. But if you look at many Middle Eastern countries and African countries, this is a way to keep fortunes within the family, is that you marry first cousins. And so mutations that are hereditary, even though those parents are heterozygous, their offspring becomes homozygous and then have autoimmunity. And if that's something you're interested in, that's exactly what we cover in the advanced immunology classes, diagnosing what is the genetic deficiency that leads to those diseases.
Okay, that is very interesting. My wife's cousin actually just had a baby a few months back that has DiGeorge syndrome.
Right,
So
and it's not always consanguineous, but the vast majority of the time it is. I think many times in the United States, it's illegal to marry your first cousin or closer. So you do still have the chance of two heterozygous people having offspring that's homozygous for mutation.
Thank you for answering it.
Sure. Other questions?
Dr. Blotman, I just had a quick question. It might be a silly question, more or less just out of curiosity. But in one of the earlier slides, it looked like a bunch of the precursors for both B and T cells. The nucleus was more or less in the center versus in the like the actual B and T cells. The nucleus was much more biased towards the side of the cell, and it would look like it was more or less attached to the membrane. Is that any significance, or is it just different pictures?
You're talking about this slide? Hold on just a second.
Mm-hmm.
This slide?
Yeah, that's the one.
Okay. So you're very adept to pick up on that detail, and that's done intentionally. As the cells are in the thymus, they're actually undergoing all of these steps, and so they're preparing to divide. They're preparing to make cellular decisions. And so they have larger cytoplasm. They're actively doing things. When a cell then leaves the thymus as a mature T cell or a mature B cell, it sort of stops having to do that. It's going to look for antigen, but it's basically not going to be preparing to divide. That's much later. And so the cytoplasm does go down. They become small, and this is originally why they were thought to be senescent cells, because they exit the thymus and then they essentially look like they're not going to do anything. And so yes, they become these small cells with virtually no cytoplasm. They're small, unremarkable lymphocytes that we now know are very important for the immune system, but at the time people thought were just kind of on their way to dying.
Okay, thank you.
Dr. Blotman.
Yes.
So the this is just in regards to like the final exam, because the provost had sent out an email saying that all final exams have, like, the final exam week was removed. Does that change our lecture schedule at all?
The final exam week is removed?
Yeah, supposedly.
I'll have to look into that.
Okay, because he had sent an email about that, and I saw that our final exams were during final exam week.
Yeah, I'll look into that and sort that out. Thank you for bringing that to my attention.
Okay.
I had not
welcome.
heard that. Other questions? Okay, if there's no other questions, I will see you all on Tuesday.
Do we have a bonus question for this lecture today?
I didn't think we did. No, there isn't one for today.
Okay, thank you so much, Dr. Blattman. See you on Thursday. Or, I'm sorry, next week.
Right. See you on Tuesday.